Two-photon microscopy. Can you elaborate on this technique and on the calcium imaging? How does this work? And why do you use two-photon microscopy to image, for example, in vivo mice in vivo?
Yeah, so two-photon microscopy is basically a sophisticated form of microscopy, which is not in essence not very different from other types of microscopy, in the sense that it relies on fluorescent proteins or fluorescent dyes that you can inject in the brain, and fluorescent proteins you can express with genetic tools, with viruses or with transgenic mice, for example. And these fluorescent molecules can be readily excited by light of particular wavelengths. The problem with regular, let's say, confocal microscopy is that you need quite some light to penetrate brain tissue, to go deeper than just a few microns, for example. And so that poses a problem if you want to do it in living material, and especially if you want to do it in a living brain through a cranial window, for example. And the other problem, and because you need to apply quite some light, you are causing toxicity, and you will have to filter out all kinds of background light that is coming back. So two-photon microscopy solves a lot of those issues in one go, because only a very small focal plane will have sufficient a number of photons basically to excite the fluorophores. And so you get rid in a way of the background problem, and the higher wavelengths that you use typically for two-photon microscopy is a bit more forgiving to the brain. It's less toxic, and maybe the fluorophores are less easily quenched or producing non-desirable phototoxic effects. That said, it's not it's not the golden bullet. It's there's still you can still make lots of mistakes with it and do things that you're not intending to do. So in terms of pathology or toxicity.
Yeah, for people who are not familiar with this concepts like confocal, etc., the thing that we were discussing is that in principle you have a focal plane that is illuminated, and when you use the so-called confocal microscope, you illuminate also the above your focal plane and below, but because of the technical properties of this microscope, you will kind of get rid of this out of focus light. However, you will not record it, but the cells will be illuminated, meaning that
there will be light going through this tissue, obviously. Yes.
But in two-photon, you have this only in one plane because of the nature again of the tool, that these two-photon have to be at exactly specific time to meet each other at this point, and the sufficient energy should be there to excite the dyes, right? So therefore, you have this kind of excitation in a certain plane, without kind of illuminating and damaging additionally cells that are above or below. And of course, the other huge advantage is that you can penetrate tissues quite deeply, right?
Yes, because the wavelengths are usually, let's say, typically twice as high as what you would do with single photon, and that usually is a bit that's more in the red spectrum, and that allows further penetration, deeper penetration. So it's not still, not crazy, but you can get a bit further than otherwise.
It's enough to address certain questions.
Yeah. The disadvantage is that you need expensive lasers to create this this dense enough packages of photons to get that excitation going.
Yeah, these instruments can go up to, I guess, million.
When
more.
you're talking about very, very sophisticated, but you 100,000, 200,000, it's not unheard of.
It's expensive, and it's also hard to operate. You have always this misalignment of the lasers, right? So from time to time,
it's getting better. Yes, I think when I was in Cold Spring Harbor with Carl Svoboda, we were still using lasers that needed some manual tweaking, and that was part of the morning would go into trying to get things stable. But over time and very rapidly, when the industry started seeing a market, I think they started to develop more off the shelf or like turnkey equipment. So nowadays it's a vacuum chamber that you don't open and you just switch on the key and it works.